Hello friends, today we are going to discuss about one of the important topic, Mendel's principles of inheritance or Mendel's laws of inheritance. Mendel's principles of inheritance. Gregor Johan Mendel for the first time conceptualized or consolidated the knowledge on the concept of inheritance. Particularly during 1850s or mid-19th centuries, biologists are unsure which biological element is responsible for the inheritance. And Mendel, through his hybridization experiments on garden peas, postulated that factors are responsible for the inheritance. Even he was the first scientist to prove that characters are inherited in an unblending fashion. Because during those days, biologists believed that parental characters will get inherited to the offspring in a blending manner. Blending means average of the phenotypical characters of the parents. Say for example, if one parent is tall, if another parent is dwarf, the mixture, that means the average of the same will get inherited to the offspring. It is a generally prevailed opinion during those days. Even the topmost biologists also believe this. That is why even for Mendel, it is difficult to convince the topmost biologists of the time. And unfortunately, he has been recognized as the posthumous father of modern genetics because it is considered as a black spot, is my opinion, because a scientist should enjoy his credentials based on his extreme hard work. And Mendel is the first person to prove that factors are responsible for the laws of inheritance. But unfortunately, no one has recognized this point when he was alive. After he died, then the scientists believed that yes, the factors are responsible for the laws of inheritance. No, we consider these factors as genes, that means alleles. So somehow, and has been considered as the father of modern genetics, and he is a reputed person in what we call in the chief architecture of the modern genetics. Okay, Gregor Johann Mendel. Belonged to current Czech Republic, and to prove his point, he has used Pisum sativum, the garden pea plant. Pisum sativum, garden pea plant, and he has done the hybridization experiments for a period of 7 years, starting from 1856 to 1863. As I said, he has consolidated or conceptualized the laws of inheritance through, that means the experimental object here, that means the plant is Pisum sativum. The laws of inheritance are considered as the principles of inheritance, and there are totally 3 laws. Number 1, the first law, the law of dominance. Number 2, law of segregation. Number 3, law of independent assortment. Here for convenience we can divide into 2 frames, monohybrid cross and the dihybrid cross. Through monohybrid cross, Mendel has postulated law of dominance and law of segregation, and through dihybrid cross, Mendel has proposed law of independent assortment. That means totally 3 laws which are fundamental base for the laws of inheritance. Later the same has been, that means the basket has been broadened by the successive scientists with respect to nature's complexities and a diverse variety of diverse varieties associated with DNA. All these are actually relied upon the Mendel's framework. That is a remarkable contribution of Gregor Johann Mendel. Okay, as I said, Pisum sativum is the plant which he picked up for his investigation, and he has picked up totally 7 characters. 7 characters with 14 contrasting traits. Contrasting traits means each character have 1 pair of contrasting traits. Each character. That means before we go into the discussion, we have to learn some basic. That means we have to learn about some of the important terms associated with this. Traits. Here character traits. Traits are nothing but they are the contrasting form of one particular character. Say a character stem height, tall is one form, dwarf is another form. That means the 2 contrasting forms of a character are considered as a trait. They say the seed color, say a yellow color and a green color. And these are trait is even one of the alternate forms of a character. One may be dominant, another may be recessive. Here, this can be correlated with alleles. Alleles means the slight modifications of the same gene. That means the two alternative forms of a gene are alleles. Just like the one allele exhibit, that means expresses one character, code for one character in one trait, and another allele encodes for another trait. So respective allele have respective traits. Therefore, one character have two shades. Just like we have one coin, two sides of a coin. Say one character have two traits. One that means here the traits are contrasting. The traits are based on alleles, and Mendel considered these as factors because he doesn't know what exactly the responsible. No, it is considered as gene. Okay. Then another term associated with here with Mendel laws is phenotype. It is nothing but the physical appearance or outward appearance of an individual. In a genotype, the genetic makeup of an individual. And homozygous if the alleles are identical. Heterozygous if the alleles are dissimilar. So these are some of the important words which will come in our next subsequent discussions. Okay. Then he has used 7 characters, and that means each character with one pair of contrasting traits. Totally 14 contrasting traits. And he has used true breeding plants. True breeding pea plants. No, true breeding is very important. True breeding means the plants which undergo self-pollination continuously so that a stable character will get inherited and expressed over successive generations. And this is very important. And because Mendel is the first person to prove that characters will get inherited from parents to offspring in an unblending manner. In an unblending manner. Unblending manner. Inheritance. Because during those days, even the topmost biologists believe that the average of the parental characters will get inherited. And he is the first person, Mendel was the first person to prove that characters will get inherited in unblending. Say if the parent is tall, the offspring will be tall. If the parent is dwarf, the offspring will be dwarf. It is called unblending. And also Mendel was the first person to prove that in the use of mathematical equations and statistics in the biology experiments. Till that means during those days there is no such culture or a system to use mathematics to blend biology with mathematics. Generally we call biometrics, mathematics with biology. But he was the first person who used mathematical equations and statistical aspects in understanding that means conceptualization of biology. And for the first time, mathematical and statistics were used, and he was the first person to prove that. And on top of it, and he has proved that factors are responsible for the inheritance. As I said, totally 3 laws, important laws, which are the fundamental basis or framework for inheritance, and they are undisputed laws. Whatever we are learning today are coming from the basics of Mendel, and that is why he has been considered as a father of modern genetics. Okay, and it's about the basics. Then I told you now there are 7 characters. Yes, pick it up 7 characters, and the plants are true breeding pea plants. And he has used large sample collection. The credentials of Mendel. Why we have to believe Mendel's experiment, Mendel's investigate experiments? Because he has used large sampling size for a period of 1856 to 67 period. That is the most important credentials. And also he has the inferences which he get from the experiments has been successfully proved in the successive experiments. That means confirmation of inferences or what we call deduction along the successive experiments has been proved by Mendel, and it is another credential, another significant aspect of Mendel's investigations. Okay. Then the 7 characters which he has used in the true breeding pea plant are: number 1, seed color, seed shape, flower color, flower position, pod color, pod shape, stem height. Now, as I said, each character have one pair of contrasting traits. One is dominant, another one is recessive trait. Say seed color. The dominant color is yellow. The recessive is green. And seed shape. The dominant is round. The recessive is wrinkled. Flower color. The dominant is violet. The recessive is white. Flower position. The dominant is axial. The recessive is terminal. Pod color. The dominant is green. The recessive is yellow. Pod shape. Full or inflated. Recessive is constricted. Stem height. Tall is dominant. Dwarf is recessive. So this is about the 7 characters which with respect to contrasting traits which Mendel has used in his experiments. That means the experiment here are true breeding plants. Okay, and here for convenience we can categorize into 2: monohybrid cross and dihybrid cross. We have discussed. Monohybrid cross leads to law of dominance and law of segregation, and dihybrid cross leads to law of independent assortment. And each have, that means monohybrid cross means the word it implied, the crosses between plants differing in single trait is monohybrid cross. For example, we'll take one particular cross. That means involve 2 plants. They are similar in all characters except one trait. Say seed color or seed shape or flower color or flower's position. That means similarity in all the characters except in one trait is a monohybrid cross. If we learn one particular monohybrid cross, that means the results he got the same in other crosses also, other monohybrid crosses as well, which involve other characters. And the same for dihybrid cross. The cross involve, that means the cross between plants that differs in 2 traits. So you can take any 2 traits. The thing is, the significant is that the results of one particular dihybrid cross are similar to other dihybrid cross also. That is why I told you now, consolidation of laws of inheritance proved by, that means Mendel is the first person who has consolidated the laws of inheritance. Okay, we'll discuss point by point how he has proved his point of first Mendel laws. The monohybrid cross, the word it implied, monohybrid cross. Monohybrid cross. He has done the experiments, that means hybridization experiment, that means cross pollination or artificial pollination of 2 different plants. That means plants differing in one trait. Say we will take one particular character, stem height. A tall plant, say parents. Tall plants. I told you already, contrasting traits are controlled by factors what we call alleles and they occur in pairs. A tall plant presented by this and a dwarf plant. Tall is dominant, homozygous dominant and dwarf is recessive. And the gametes comes out of it. Gametes. Then we got the F1 hybrid, which is again a tall. He has subjected the F1 hybrids to selfing, what we call F2 generation, to see how it will come. Say, for I will tell you the Punnett scale we have to draw to understand the phenotypes and genotypes. These are the gametes, female gametes and male gametes. So based on this, the F2 generation leads to the phenotypic ratio. The phenotypic ratio is 3:1. 3 are tall and 1 is dwarf. And genotypic ratio 1:2:1. 1 homozygous dominant, 2 heterozygous dominant and 3 dwarf. Okay. this is what he got. That means the two different plants that mean different in one character, tall and dwarf. One parent is completely tall and dwarf. We got a F1 hybrid. On selfing we got phenotypic ratio of 3:1. 3 are tall and 1 is dwarf, and genotypic ratio 1:2:1. 1 are homozygous dominant and 2 are heterozygous dominant, and 1 is dwarf. Here the point is this is very important and key to understand. You know, the tall, the F1 hybrid actually a heterozygous. Mendel later found that in heterozygous F1 hybrid, though there is a allele, that means it is an allele for dwarf and this is allele for tall. We have discussed. Respective trait will be controlled by respective allele. Okay, and though there is an allele for dwarf, but tall has been expressed because tall allele, that means capital T, dominates small t. This is the first law, what we call law of dominance. Law of dominance. The first law. That means according to the first law of dominance, characters are controlled by discrete units called factors. Now we consider it as genes. And these factors occurs in pairs. These factors occurs in pairs. Out of it, in a heterozygous condition, one character will dominate the other. One character, that means the character which dominates the dominant trait, that means dominant allele and dominant trait, and which has been, and the other is called recessive allele and the recessive trait. The same has been done. And the law of dominance also explains one of the parental character in F1 generation, whereas both the parental characters in F2 generation. It also explains the 3:1 ratio. That means both just only either one of the characters will express in F1 and both the parental characters in F2. So here the point is simple. In a heterozygous condition, one allele is dominant. That means so as to express its trait, dominates the other trait. So it is called law of dominance. The word it implied. It dominates the expression of others, so that only the expression of one particular parent will get expressed, whether other parent remains recessive. This is called law of dominance, the first law. Okay. Then the second most important point, what we call law of segregation. We have to look at the F2 generation. In F1, we didn't get any dwarf varieties, only the tall varieties we got. Even a Mendel doesn't know that, what is responsible for this. And he was surprised when he sees some dwarf varieties. Because the character which has been suppressed, which has been recessive in F1 was recovered in F2. It is the first surprise that Mendel got. How come that dwarf which has been become suppressive or recessive in F1 become recovered in F2? So he believed that something, a stable feature has been inherited from one generation to another generation. Stable, I said, without unblending, in an unblending manner, without blending. Without blending is most important. I told you, during those days, biologists believe that the average of the parental characters will get inherited. But unlike that, the generally prevailing idea, he got the results. That means if the dwarf parent gives to dwarf offspring, the tall variety gives to tall offspring, there is no mixture, there is no admixture. And since here in F2 plants, there are some plants which are dwarf. That means he believed, based on it, he deduced a point that alleles, we knew the gamete formation, gametogenesis. Generally during gametogenesis, the gametes will receive respect to allele. That means the alleles will separate. Here alleles will segregate from each other so that each gamete receive one allele. This is very important. I repeat. Here the alleles will segregate from each other during gametogenesis so that each gamete will receive one allele. The same will get, it's like waiting for an opportunity. Just forget about dominance and recessive. Just the respective allele, respect to recessive allele, I am saying, it waits for its opportunity to express without blending or unblending. So that is why in F2 we see the dwarf varieties, which has not been seen in F1. That means a stable, that means the alleles are segregated from each other. There is no admixture. Segregate and it gets respect to expression. That is very important because in F2 we got dwarf variety. That means alleles segregate from each other during gamete formation so that each gamete will receive one particular allele. This is called law of segregation. Second law or law of segregation. This is very important. And the first law is based on this concept, and the second law, is like based on how, based in F2, again the dwarf variety, that means the recessive variety has been recovered. So based on this, he has propounded the second law. And I told you, checkerboard, Reginald C. Punnett, a British geneticist, developed this Punnett square, what we call Punnett square. That means it is nothing but the graphical representation to calculate the probability of possible genotypes in offspring. We have it's about possibilities when 1:2:1. We are giving a prediction regarding the possible outcome, possible genotypes of offspring. 1 complete homozygous dominant, 2 heterozygous dominant, and 1 recessive. This is what exactly the beneficial aspect if we use then a Punnett square propounded by Reginald C. Punnett. Okay, so based on this monohybrid cross, he has proved the first law of dominance based on which dominates other, and based on its respective character, and the second law of the law of segregation, because alleles will get separated during gamete formation, so that each gamete will receive one particular allele. This is very important. Okay, this is second law of Mendel, or law of segregation. And most important feature is we have given the prediction of genotypes. That means prediction of genotype, but not the genotypic composition. In monohybrid cross, we have given the prediction with the help of Punnett square, the possible outcome of genotypes in offspring. But what is the method to exactly identify the genotypic composition? Say, with respect to particularly this case emerges when there is a tall plant. We don't know whether it is homozygous or heterozygous, and how would have to assess whether it is heterozygous or homozygous. So Mendel has worked out another what we call test cross. Test cross. In a test cross, the dominant phenotype will be crossed against a recessive parent to identify the genotype of the dominant phenotype. Say the F2 tall plant, we got it now, 1:2:1, 2. The one homozygous dominant, 2 heterozygous dominant. We know now, but during those days the people don't know. Even Mendel also. What exactly the composition, whether it is homozygous or heterozygous. So to ascertain the same, he has used test cross. And in test cross, the F2 tall will cross against the recessive parent, that means dwarf. So that say we will say, for example, violet color flower, the dominant phenotype. Say dominant phenotype. Dominant phenotype. Say the violet color. That means flower color. Flower color. Okay. There are two possibilities here. Either heterozygous or homozygous dominant. Say the dwarf variety. That means white color. Small w, small w. It have two gametes. Here are the same. White color. There are two varieties. Small. This is a recessive trait. Okay, we all discussed. Violet color is flower dominant and white is recessive. And dry checkerboard, a Punnett square. The same concept here. Heterozygous dominant, and recessive. And here are the same concept. Here homozygous dominant, This is homozygous, and of course this is recessive. These are the respective genotypes. Here we got here in first cross, that means when the condition, when the first case, when the dominance is heterozygous, we got 2 violet colored flowers and 2 white color. That means half, 50:50. But here all are violet. all are dominant. These are result. That means in one case where there is a homozygous dominant against the recessive plant, we got all violet. So therefore interpretation is homozygous dominant. Homozygous dominant. With respect to the genotype of the dominant phenotype involving in this particular cross, particular test cross, having a genotype of homozygous dominant. Because all are violet. It is possible only when there is homozygous dominant. That's interpretation. This is very important. Violet color, all violet because if it is homozygous dominant. And here, it is heterozygous condition. Here, the 2 are violet and 2 are white. Because here, since 2 are white, that means recessive, it is possible when there is a heterozygous condition. So, the resultant leads to, that means the test cross proves the composition, that means genotypic composition. Genotypic composition. This is very important. So how? So based on test class, one can identify the genotypic composition of a particular dominant phenotype, say F2 tall, for example, or we have to take the example of violet color. Okay. Next, dihybrid cross. Now this is very important because he has propounded the most important law, the third law of independent assortment. Dihybrid cross. The word it implied, crosses between plants which differ in 2 traits is a dihybrid cross. Say, we can take 2 particular examples. Say the parents, round yellow, which are homozygous, okay? Round yellow, and wrinkled green, here wrinkled green, okay, parents. We got the gametes, respect to gametes, capital Y, and the same we got it. Then we got ultimately the F1 generation. Again it is round yellow. F1 generation. Okay. The same concept as we follow in the monohybrid cross. The same is subject to selfing. Say we got it now. The 4 different gametes we got. I will tell you how it ends. Okay. The 4 different gametes we got. So these are the parents and gametes. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Totally 16 you have to get. 16 boards. 16 we got. The respective genotypes are like this. It is very easy to understand once we understand the logic. Or capital Y. Then R. Then so totally 16 possible genotypes we get. R then Y. R Y Y. R Y Y. R Y Y. Then the phenotypic ratio, so we got 16 type of possible genotypes, and the phenotypic ratio we got is phenotypic ratio we got is 9:3:3:1. In the sense 9 are round yellow, the phenotypes, 3 are round green, 3 are wrinkled yellow, and 1 is wrinkled green. And the genotypic ratio is 1:2:1:2:2:4:4:4. 2:1. Here, the most important point is we have to discuss one by one for better understanding, because Dihybrid Cross leads to most important law of independent assortment, our third law. You know that we have discussed the crosses between plants which differ in two traits is a Dihybrid Cross. What are two traits? Say round and wrinkled. That means they are nothing but seed shapes, and yellow and green. They are nothing but seed color. Here round is the dominant trait. Here yellow is a dominant trait. Wrinkled and green are recessive trait, and their respective alleles. Capta, caploi. For we use that small, and we got it gametes. And the F1 generation, as usually, we got round yellow because round and yellow are the dominant alleles, dominant traits. We got though heterozygous. The same we got subjected to Punnett square. We got 16 genotypes, out of which the phenotypic ratio 9:2, 3:3:1. 9 are round yellow, 3 are round green, and 3 are wrinkled yellow, and 1 is wrinkled green, and it is respective. It is genotype ratio 1:2:2, 1:2:1:2, 1:4:2:1, 1:2:1. Okay. The thing is, what exactly the Dihybrid Cross says, that means respect to law of independent assortment. The law of independent assortment based on Dihybrid Cross observation states